Hi class, it looks like it's about time to start. I'm glad you're all here. I realize some people cannot be here, and so hi, online people. Just as a reminder, I realize Robin probably sent you an email. Did he send you an email? I will tell you. I'm sure he's going to send another one. I'll start with this class. This class, we have our exam a week from today. Okay? So, a week from today, we have our lecture exam. I appreciate all of your excellent feedback and suggestions. From looking at that and trying to figure out what works, it looks like the only really fair way to do this is to have everybody be online. So it's going to open Friday morning. It will close Friday afternoon, early evening, might as well keep it open all day. It'll open by midnight, okay? So it'll be open all day Friday. Once you open it up to take it, you have an hour. It's open book. Question, does open book mean easy? Don't even worry about studying. No, why not? I know, because sneaky professors think, well, it's open book. They should just write. The time is going to be a little bit tight, okay? The idea is like, yes, you can use your notes, yes, you can look stuff up, but the idea is, the assumption is that it's based on the idea that you'll read the question and answer it, not frantically flip through your book being like, what? Okay? With that, it's open book, open notes. It's not open buddy. Okay, so if, and there are ways to check, if you and the person who's also in the class with you have very similar answers, like concerningly so. If you both say the mitochondria for some answer that's the one, don't freak out and try to like make it sound different, okay? But don't copy, okay? I'm trying to figure out if I should actually have different questions amongst different versions of the exam. I'll depending on how ambitious I am over the weekend, there might be different versions of this exam. Same types of questions, but like different orders for the... I'll definitely put different orders for the questions, but we'll see how exciting it gets. Fair enough? So that's a week from today. So next Friday you don't come in here. Instead you stay wherever actually has internet, just not here, and you take that test. Fair enough? If you take it during the time of class, which is what I would highly recommend, I'll be standing by my computer, I'll be sitting by my computer, and you can like shoot me emails like, what does this word mean? If you take some other time, that's on you. Alright? Similarly, you've got lab exams this coming week. Apparently I can't do words, but this coming week is are your lab exams. Again, normally you'd go in and like poke stuff and have to fiddle with a microscope. Because there's a lot of you out, the only fair way to do this is to have online for everybody. So it'll be a bunch of pictures, like can you identify this? What kind of movements does this organism have? Is it multicellular? If I want to find protein in a solution, if I want to check for protein, which of these reagents would I use? That kind of thing. Fair enough? That is going to be open, I believe, starting Tuesday, all the way through Friday about noon. That's just one, I think. The idea is you'll take it during your lab chunk. If you take it during your lab chunk, your friendly lab instructor will be there online to like answer chat questions or email questions. If you take it some other time, again, you're kind of on your own. Fair enough? Concerns about that? Great. And then you have a quiz today, so take that sometime before tonight. We'll get out early so you can do that. I feel like I spend a lot of time telling you administrative stuff, but this semester is weird, so I'm just going to keep doing that. Any administrative type of questions? Okay, on to the cell. This is the last day that we're talking about just the cell proper as like what are its chunks. Starting next week, we start moving on to other things. Chunks that we haven't seen yet or haven't talked about yet are energy-related portions. Chloroplasts and mitochondria both can change energy from one form to another. And we'll spend a week on each, actually. Um, Mitochondria are where this, the cell is going to make ATP. It's going to use, say, sugar, break it down, and use that to make ATP, which can then be used in the cell as an energy form. Fair enough? More on this later. Not to worry. So much more on that. It's so beautiful. Chloroplasts are going to be sites of photosynthesis. So that's going to take energy from the sun and use that energy to trigger a series of reactions that produces like sugar and stuff. Okay? The chloroplasts and mitochondria are both kind of weird. They're cool, but they're weird. The best guess for how cells happen to have them is this. It's called the endosymbiote theory. And the idea is, once upon a time in the distant past, there's the ancestor of a eukaryotic cell, right? It's got a nucleus there, it's got some membranes, it's beautiful, right? This cell presumably gets its energy by engulfing other things and eating them, right? So it tries that, and it tries to gulp a prokaryotic cell. But instead of this little guy dying, it ends up happily living there. And it lives there, has a nice, warm, cozy environment, is protected from other things eating it because it's already been eaten, right? As it receives sugar and other things that the cell happens to have lying around, it produces ATP, and this cell thinks this is a really good idea. Both of these survive better together, right? We don't know for a fact that that's how that happened, right? Nobody was around there with a microscope watching it, but it seems really likely because mitochondria behave kind of like a little tiny cell inside a cell. They have different DNA from the rest of the cell, which is weird, right? And like when people are made, when two people love each other very much. Anyways, when babies are made, I have a whole spiel about this too. But for this class, when egg and sperm come together, right? The father provides half of the set of chromosomes, right? The mother provides the other half, right? And same chromosomes, but different versions, mother and father, both, right? Mitochondria, you just get from your mom. So there's a slight exception to that, but for all intents and purposes, you just get from your mom. So nuclear DNA from the nucleus, half from your dad, half from your mom. But mitochondrial DNA you get because the egg still has the mitochondria inside of it, and that's the mitochondria you get. And they do their own thing. So if people are looking for like, family history things, looking at maternal mitochondria is often the way to go because it's just straight maternal, right? And it doesn't have any of that weird mixing stuff, good mixing stuff that happens for the nuclear chromosomes. Anyways, mitochondria, different, weird, for all the world, kind of look like a little tiny organism inside a cell. They're not their own organism. They can't live on their own now. If you pull mitochondria out of the cell and just have it in a dish, it's not going to form a thriving colony, right? It's now completely dependent on the cell. But it's still kind of weird, and it's got this nice membrane structure. It looks like a cell. Chloroplasts are similar in that they're also kind of this cell inside a cell idea. And yeah, the idea is some sort of photosynthetic prokaryote. There are photosynthetic prokaryotes. Got engulfed by a eukaryotic cell, and then they had both. In something like plants, you're expecting chloroplasts. You're also expecting mitochondria. Sometimes people think like, well, if you have one, you can't have the other. You can't have the other. You don't have chloroplasts, but plants can have both. Fair enough? There's actually, I don't even know where this is at. There's some, I hesitate to say harebrained, but that's idea of like, what if you could inject chloroplasts into your skin and then like charge in sunlight? I have no idea where the state of that currently is. But stay tuned. Concerns about this? Okay, So, mitochondria, lots and lots of words. What that's trying to say is mitochondria show up in almost all eukaryotic cells. Not every single one, there are a few exceptions, but pretty much eukaryotic cells are going to have mitochondria. They've got an inside and outside membrane. They've got a lot of surface area. So don't memorize all the words, but got mitochondria, layers, and lots of membranes. Do you see that? Lots of surface area. This is going to become important when we figure out what this is actually doing and how it's doing it. Chloroplasts are the answer to when your little cousin asks, why is grass green? That's going to be the answer, if you want that to be the answer. Or if you want it to be like, because it's beautiful. That's a fine answer too, depending on how old the kid is. Chloroplasts have a pigment called chlorophyll in them, and they've got other things too, but they can capture light energy. So they show up in plants, they show up in algae. It's this organelle like this. You'll have a lab doing all sorts of things with chlorophyll, with like sucking it out of spinach. I mean, not physically, but grinding up spinach and like pulling out chlorophyll and looking at it. We're good? Okay. So those are the last organelles I wanted to show you. The final component of the cell proper we're going to look at today is called the cytoskeleton. It's the stuff inside. All right, so everything's inside. Okay, it's the structural support inside. So people have this idea that a cell's like a bag of water, which is mostly true. But it's a bag of water with a lot of internal supports. It's got a whole series of fibers. And so here's a picture. This is a neuron. It's stained for the cytoskeleton, both green and red. This is the nucleus. But do you see that there's a lot of cytoskeleton there? A lot of these fibers. It's holding this whole thing up. The cytoskeleton organizes where different components of the cell are. It holds things still. It lets things move. We'll see lots of things it does. It's got three types. They're microtubules. They look like tubes. They're microfilaments. They look like little tiny strands or hairs. And then there's intermediate filaments. They're halfway between both of them. And there's a little video to watch here, just of, because it's cool. Cytoskeletal elements, this is the YouTube video that's linked here, are going to be growing, and as that skeleton grows, it's not like bone, but as that internal structure grows, it pushes the edge of the cell forward. So this is a neuron that's trying to grow forwards. And it looks like it's crawling, right? It's because those tiny components are growing forwards and dragging the cell with it, basically. That's okay? We're cool with this idea? All right. So cytoskeleton is both doing support, right, holding everything up, and also motility, the ability to move. It's going to interact with things called motor proteins, which are super awesome. I have a dumb video to show you. I inflicted this dumb video on the 9 o'clock class and asked them if they were scarred for life, and they said yes, but that you had to see it too. So it's this YouTube video here. It's factually accurate, but creepy. We're not going to watch it all because, we have other things to do with our lives. It's the one that's linked there in the bottom of the screen. Can you hear it okay?
Our brain is made of billions of nerve cells, and they're all connected. If we take a closer look, a nerve cell seems to have antennas. Most of them are receivers of information, but only one is a transmitter, called the axon. This axon is connected to several receivers of other cells, forming a gigantic neural network, the brain. Meet John. John is a kinesin, a motor protein. He lives inside a nerve cell, and he has a proper job. To ensure that a brain cell does his job properly, it needs the continuous flow of building materials, proteins. They travel through the cell using the cytoskeleton. If you would compare a nerve cell with a city, the cytoskeleton inside the cell would be the roads, and the traveling proteins would be the traffic. These materials are towed by motors along the roads. And just as in real life, there are different kinds of motors and different kinds of roads. John's sole purpose in life is to deliver his cargo to a specific place in the axon. He takes the main roads, and he walks in just one direction only. John's job may seem easy, but it's not. He has to overcome a number of obstacles to ensure that the right amount of cargo arrives at the right place. To make the journey even more difficult, John is not alone. Other motor proteins ride along with his cargo. They haven't woken up yet, but that will happen soon. The journey starts in the center of the city, just like in the center of the cell. To enter the axon, John has to pass a place called the axon initial segment. In this segment, there are two kinds of roads. The main roads that John uses, called the microtubules, and a lot of little alleys called actin. And here, our brave motor protein meets his first challenge, because one of his sleeping travel companions, myosin, has woken up and starts to cling to the actin. And there are a lot of alleys. Only brute force can save John now.
John makes it, just so you know. Thoughts? You like it? Okay, so like, I do, but I don't know why. Like the production quality is really good, but they also put eyeballs on the thing, right? Anyways, okay, but we're okay with the idea there. And these motor proteins, they legitimately, minus the eyeballs and the fuzzy slippers, they look like our buddy John up there. They've got little legs. It costs energy to do this, but they walk along microtubules. They legitimately walk along, which is important. Here's a microscope picture of that. You've got these vesicles here, microtubule here. There's a motor protein down right here that's walking along. And it's important because moving stuff around cells you need this to function. So like, for instance, some cells are really, really big. A neuron that like goes all the way down to your toe, so if you drop something on your toe and you feel it, the cell body of that neuron is living in your spinal cord, right? That's, depending on how tall you are, is a long ways, right? So you need to be able to pack a vesicle full of proteins or whatever it is you're packing it full of, and then attach it to protein and have it walk on down. And the cytoskeleton provides that road. Questions? With that, this is just so biology makes more sense in the future. You'll see the squid giant axon used a lot as a cell model, okay? Called the squid giant axon. This thing is huge. You can see it with your naked eye. Like, it's small, but it's like as thick as a hair. So from a cellular perspective, it's ginormous. And biologists love this thing because it's easy to find and it's easy to get. Now, when I was an undergrad, I heard about the squid giant axon, and I had this mental picture of like, it's a giant squid and you get the axon out of it, and I had no idea how biologists did this. Like, how do you get a giant squid and get their axons? They're actually these little buddies. It was sort of disappointing, but It's the giant axon of little squids. Not giant squids. Anyways, just so you know, in case the world needs to make more sense. It was kind of disappointing. It really was. But also cleared a lot of things up. Just so if you see that again, that's what we're talking about. But on to the cytoskeleton. 3 main parts, and I saw that you all were taking notes earlier. Microtubules, microfilaments, and intermediate filaments. Microtubules are the fattest ones. They're thick. They're tubes. Microfilaments, they're also called actin filaments. So most people are coming at this from biology proper, typically call them microfilaments. People coming at this from the anatomy, physiology, health sciences usually call it actin. It's the same stuff, just different names. They're really thin, and then intermediate filaments are kind of halfway between those two. Microtubules are legitimately a tube. They're stained green here in this neuron. It's a polymer of something called tubulin. The naming system, I know, they're like, great, we'll call it tubulin. It's made up of these tubulin dimers, and then just forms this coil. Okay? This is reasonably strong, and it keeps the cell held up. It's strong. It's also able to have things walk along it, and motor proteins can interact with this stuff really well. They can interact with it, and what can happen is you can have. Oh, I remember. I'm going to show you pictures, and I'll talk to you about why these are cool. So, microtubules, the tube, small but the biggest of these options. Yeah? Microfilaments are 2 little tiny intertwined strands, again a polymer, built up of repeating subunits, really thin. And then the middle one, the kind of the well-adjusted middle one, are called intermediate filaments, not as small as actin, not as large as microtubules. Microtubules and microfilaments both are going to do a lot of support, but they're also going to do a lot of movement. They're going to be involved in physically moving the cell, or contracting it, or spreading it out. Intermediate filaments don't do either of those things. They're going to do support, but they're not worried about moving the cell that much. So far, so good? The structure is the same general structure as, have you ever had that like Twizzler candy, those licorice things you can pull off a strand? It's that picture. Yeah. We're solid? Okay, so microtubules, hollow rods, really easy, like they build really fast, and they break down pretty fast. Shape of the cell, again, cell movement. If you've seen, I drew it for the other class. Well, I don't know what these childhood fears over here are. If you've seen this picture before, during cell division, where you've got chromosomes, and then there's something coming and it's going to pull these apart. This stuff, the stuff that's pulling it apart, those are going to be microtubules. Now, microtubules are going to be able to interact with motor proteins. What can happen is, because a motor protein can grab a microtubule, it can grab and it can basically pull. If it pulls and releases, you can get it to flap. So if you have something like a flagella, you can have the interactions with motor proteins and this microtubule propel movement forward. You can have it for like little cilia, the like little hair-like things. They can move as well. Similar idea. Cytoskeletal structure being interacted on by proteins. Yeah? So that was the big one. We're now looking at the smallest one, the intermediate one I saved for last because it's the most different. Microfilaments are solid. there's actin. They're not hollow in the middle. They're going to provide some 3D structure as well, and they're going to also be involved with little microvilli, little guys up here. There's actin right here, intermediate filaments down there. Are people familiar with actin at all? Does anybody have any body system they associate with? No? No exercise physiology people want to leap in with this? Actin interacts with something called myosin for muscles. Yeah, okay, so some people have seen this before. And it looks like this. So you've got actin filaments, you've got myosin filaments, and you've got these cytoskeletal elements, and they're attached. And then they can walk towards each other, right? They can pull, and this causes contraction. So this drives muscle contraction. Comfortable with this idea? Since you've got the cytoskeleton anchored to the cell, if you move parts of it closer together, you can change the shape. Fair enough? So having localized movement of actin and myosin can make a cell contract. It also can make amoebas move, can have amoeboid motion. And cells other than amoebas can have what's called amoeboid motion. It's this kind of like swooshing forward motion. Just for the record, Robin is going to be writing this exam for your lab exam. Every Gen Bio lab exam I've ever seen him write has some version of a question like, how do amoebas move? Right? The answer is amoeboid motion or using pseudopodia. I mean, there's some version of this, right? Like they squish themselves forward. And we need to see this because it's beautiful. We're not going to see all of it because it's long. I happen to like this channel, by the way. We've got this amoeba. The cytoskeleton's growing out on this side, growing out on this side, and so the cell wall is being shoved with it. Actin is breaking down the other side, and so it's just basically transporting the whole thing forward. It's strangely calming to watch, isn't it? It's kind of pretty. Okay, we'll see one more and then be done. Go, little buddy. That's just pseudopodia, the little sticky out bit. We're comfortable with that? Great. So the cytoskeleton is going to set this up. So yeah, you just grow the cytoskeleton forward, and then the rest of the cell gets dragged with it, and you break the cytoskeleton down behind it. So cytoskeleton sets up movement, like large scale movement, right? It sets up, you can have a flagellum move, you can have muscles contract, you can pack a vesicle full of stuff and walk it along down to the end of something, right? The cytoskeleton's also going to help move stuff inside the cell. So there's lots of good stuff inside the cell. There's going to be nutrients, there's going to be salts, there's going to be little proteins, there can be enzymes that are important for things, there's going to be nucleic acids that are floating around doing stuff, right? Like lots of stuff. And instead of having to have a special transporter for every single tiny chunk of stuff, one of the ways the cells can solve this is basically just to slosh the inside of the cell around so the stuff moves. Does that idea make sense? Just like, so long as your bathtub water keeps moving around, your little duck is going to make it the whole way around, right? I don't know if you have ducks. I don't know if you have bathtubs. Anyways, that's the idea. So what it's called is cytoplasmic streaming. Cytoplasmic meaning the cytoplasm of the cell, right? It's just going to speed up stuff moving throughout the cell. In plant cells, it's actin myosin that drives this. But you want to see a picture. it's the YouTube link that's there online, people. So we're looking at little chloroplasts moving, but there's nothing external pushing them. This is just under a microscope slide. But our cytoplasm's streaming. The cytoplasm's been pushed, and it's going to keep things moving around. Can we see that okay? The concept makes sense for us? It's beautiful. So it's mostly microfilaments actin here driving this. Okay, I will get you out in time to work. And then intermediate filaments are the ones that are neither super big nor super small. They're not doing the motion part so much, they're just doing the structure part. They're going to help support the cell and also hold organelles in place relative to each other. Questions about that so far? That's it for the cytoskeleton. And now with the cytoskeleton, we've got a whole bunch of stuff inside the cell. We have the cytoskeleton, we've got the Golgi, we've got mitochondria, we've got the nucleus, right? We've got ribosomes, we have all sorts of things. And I know I behave. I know I behave as if the cell is the end-all be-all of all things biology, because it kind of is. But the cell doesn't exist only by itself. Cells can have things on the outside of them as well. So there's going to be things that the cell makes that it then stuffs to the outside of the cell. So it's no longer inside. That plasma membrane is the boundary, but it pushes things outside for helpful reasons. Most cells do this, and these extracellular functions do a lot of different things. They do receiving signals, they'll help with sending certain signals, they'll hold things together, they'll do lots of things. Plants have a very, very prominent type of this. It's called the cell wall. So plants and fungi and some eukaryotic cells, but they've got to be single-celled organisms to do this. Prokaryotes do this. They make a cell wall. So they got the plasma membrane, right, their cell, but then they squirt stuff out. It's basically carbohydrates. It's cellulose. It's basically stiff sugar, and some proteins and some other things. But they basically spit this stuff out of their cell and now have a case around the cell. So if you have a phone, it has an inside and outside, right? Just like because it exists. But you think to yourself, it would be better if I put a case around this just to be protected. Yeah? Okay. It's the same idea. It's going to help hold the shape. It's going to be protective. It prevents too much water uptake. We will talk about this more next week when we talk about membranes. But does anybody know what happens if you, say, you've gotten injured, you go into the hospital, and instead of running saline solution in on you, like into your veins, they run pure water in on you? What would happen? So it would kill you, yeah. Yeah, it'd be very, very bad, right? The reason for that is your cells would pop. They just take up water, like your blood cells, right? They keep taking up water, and they'd burst. It's a bad deal. This is why they don't do that, right? It's not an option. But you might have noticed, like, if you drop a carrot in a glass of water, it doesn't, like, explode, right? Okay. Plants with cell walls can take up water, and the cell will expand, but it'll get to the point where it hits the cell wall, and the cell wall will push back. So it can fill up with water, and at a certain point, it can't take on any more water, and it's just held. We okay with the idea? This is very helpful for small organisms that are like single-celled organisms, say living in fresh water. This helps make life better for them. You do not have a cell wall, but you do have stuff on the outside of your cells. So animal cells are going to have something called the extracellular matrix, which is a fancy way of, so extra in biology is going to mean typically outside or in addition to, right? Like Like, you can imagine when you hear the word extraterrestrial, it means outside of Earth, beyond Earth. This is outside or beyond the cell. And this matrix just means it's a bunch of stuff. It's basically protein and sugar. It's the stuff called glycoproteins, things like collagen, stuck on the outside. And then there are little proteins here. They're called integrins. They're integrated into the membrane. You can start to get a sense of the naming conventions, right? People just make up names with things they think make sense. These proteins are integrated into the membrane. They're integrins. They're holding on to all this extracellular matrix stuff, and they can hold on to stuff inside. So they can anchor the cell relative to other things. Does the idea make sense? You can see it here. And this whole lot of stuff is really helpful because extracellular matrix now is outside the cell, right? It can interact with the wide outside world. So mechanical changes to this, like push or pull or squish, that can be translated through, and the inside of the cell can notice it. This can be hanging around on other cells. It can sometimes, whole series, but sometimes changes here trigger different genes to be expressed inside, just because it's got a link to the outside world. Questions? Oops. That's all that. Finally, or mostly finally, cells. So they can be connected through like their extracellular matrices, right? The goo on the outside of one cell and the goo on the outside of the other cell can be next to each other. Or cells can actually physically touch. When they do that, that's what's called gap junctions. So two distinct cells, they've got different nuclei, but they're physically connected by these gap junctions. Right? Like cardiac cells do this. Unique cells, but they're continuous with each other. They've got holes opening up between them. Everyone's happy with this? Okay. Two things before we go. One brief philosophical yapping, and then a clicker question. Philosophical yapping. The cell chunks are great. The little parts, super important. But each one by themselves, just a little petri dish, is not very cool. It's when they all come together, they do something more than their individual chunks. So like macrophages, it's a cell that can go around destroying bacteria. Great. You need lots of things. You've got to have like the cytoskeleton that can move and support this. You need to have the plasma membrane be able to adjust in real time. You need to have lysosomes that can destroy this bacteria once it gets a hold of it, right? Any one of these by themselves doesn't mean much. Fair enough? Okay. On to today's one and only clicker question. And I really hope we're solid on the answer. If not, this is why we're doing it. Oh. Once it's read yours, you can put it down. Okay, anybody who either didn't read or just forgot theirs, let me know, write your things down really quickly. So I can take a sip. Anybody who's here who was on that list? Raven, anyone else? Remind me your last name? Perfect. Okay, is that everybody? Sweet. Yeah, see you on Monday. Avoid the plague over the weekend. All the things. Remember your quiz. Do remember your quiz.
I did.